from Micro TV. This is TWiEVO. This week in Evolution, episode number 99, recorded on February 27, 2024. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent, great to see you. We are in the middle of a podcasting frenzy right now.
Indeed.
So we weren't, it wasn't that long ago since we were last live streaming, less than 2 weeks. And I guess it's sort of a confession at the top here. We're stacking the numbers in our favor to line up to triple digits, TWiEVO 100, which is our next one after this. We've been furiously podcasting to get here so that we can time this all out.
Yeah,
but great.
Let's give some information on that. Want to do that
Yeah.
right up top here?
Great idea.
We'll do it twice because there are only 28 people here. But here we have a little flyer.
Yeah.
TWiEVO 100 live at the Science Mill in Johnson City. This is Sunday, March 10th at about 5 p.m.
Mm-hmm.
If you'd like to come, there'll be a little reception afterwards with food. You see there, RSVP at microbe.tv. Some of you may be around, so come by. I know that Neva, who often comes, wants to come. So just give us an RSVP, only because we want to know how much food to get.
That's exactly right. So Johnson City is a small town, and a lot of the restaurants are closed on Sunday, but we've lined up some catering. You can join us for TWiEVO 100 in vivo at the Science Mill. The Science Mill is a great place to check out whether TWiEVO is there or not. And so it'll be just after hours. The Science Mill closes at 4. We'll be scrambling behind the scenes to set up for our live streaming podcast, celebrating 100 episodes together. And then, as Vincent was saying, dinner is served afterwards. Very informal, but it'll be nice. And we'd love to connect in person, compare notes, and celebrate this moment in our podcast's sort of the descent of TWiEVO over now. Are we closing on in 6 years, or we're beyond 6 years, Vincent?
Yeah.
We've been doing this for a minute. Yeah.
Because you had we started in 2016, maybe 20, because I know you got the award at ASV in 2015, right?
Mm-hmm, mm-hmm.
That's when I first mentioned it to you.
Yeah.
So, over 6 years.
Yep.
All right. So RSVP at microbe.tv if you'd like to come. And we'll have a guest right now, so to be named.
We will, yeah. And we'll be celebrating the Science Mill. We've been talking about spaces that celebrate science. This is one in person, and we'll be talking to some of the folks there.
Oh, that'd be good.
Well.
Have them talk about the Science Mill on TWiEVO. That'd be great.
No, exactly. And I think my idea, we're kind of workshopping this in live stream, which is sort of our style here, is, so I still remember, Vincent, when you were here in Salt Lake City, I can't remember how many years ago this was, before the pandemic, and we did a live stream pre-YouTube days, but we did a podcast with some of my colleagues here as part of their microbiology and immunology retreat. Yeah. And you and we spent some time together at the Natural History Museum in Salt Lake City.
Yeah, it was cool.
Yeah. And I still remember we were kind of walking through and you say, what you asked me, said, what's missing from the Natural History Museum? And I said, I don't know, like there's not enough bathrooms but that's not the case. That's and what your answer was viruses. We're missing viruses.
That's right.
When we look at science museums, when we step through spaces that celebrate science. And so I think we might workshop that conversation actually as part of TWiEVO 100. With some of the folks at the Science Mill and talk about some of the fun things they're doing, thinking about viruses and putting that back into a Science Mill setting. So, yeah, stay tuned. If you can't make it in person, so I should say Johnson City, Texas, about an hour west of Austin. If you can't make it in person, about an hour northeast of San Antonio. But if you can't make it in person, we'll be live streaming on all channels, and please join us to celebrate, because I think it's just going to be really fun to be together and to mark this 100 episodes, Vincent. That's not shabby.
It's good.
I don't think I've done 100 if I count the number of things I've done 100 times in my life. That's a pretty small list.
Yeah,
And so
indeed.
this feels like a fun moment
So,
to celebrate.
once again, here's the flyer. RSVP at microbe.tv if you're going to be in the area.
Yeah, we swapped positions in the flyer from our usual live streaming arrangement there. It's good to mix it up. Yeah.
So thanks everyone for coming today. I want to thank our moderators. Les is here. Barb Mac UK is here. And Andrew is here from New Zealand.
Great. Welcome all.
Welcome. Thank you very much. And where are you all from? Let's have a shout out.
Yeah.
Where are you? Let us see.
Got a shy crew that's joined us. We are.
Claire is from the UK. Hello,
Great.
Claire.
Hello.
Jeremy's in Seattle.
Great.
That's cool. I know Abdulaziz is from Saudi Arabia.
Welcome.
Very cool. Always here. Thank you so much. Barb Mack, of course, is in the UK. Emmy Wheeler is in Helsinki. Dark and wet, -1C.
Whew. Sauna weather continues.
In New York, it is 12C and cloudy. It's a little warmer. It's good. Jan is from Tucson, Arizona, 17C.
Ooh, that sounds nice. Yeah.
And Andrew, of course, is from New Zealand. John is from Minneapolis. I know that.
Fantastic.
Levis, I remember you from Colombia. Philip, I don't remember where you're from. Tell us where you're from, folks. Calliope. Calliope Nikitas. What a cool name. Is that really your name, Calliope? That's a great name because the Calliope is some kind of a thing in a circus that makes music, right? Toronto, welcome. Sean is from Tennessee.
Great. Welcome.
Camila is from Germany. Joseph is from Ontario. Yeah, there's John. 44F in Minneapolis. Yeah, hey, MJ, greetings from Austin, Texas. Well, hey, MJ, you should come check us out Sunday. What was the date again?
March 10th.
March 10th.
Yeah, a couple weeks out. Not too far off. A little less than 2 weeks. It's happening. Yeah.
Sean is from New Jersey. Elizabeth is from West Virginia.
Hello.
And this name that cannot be mentioned with pizza at the end is from Virginia.
That's great.
And that's, let's see, Calliope is one of the muses. Yeah, it's correct. But it's also some kind of instrument, right? Let's look it up. Calliope is, it's one of the muses. Oh, I thought it was a thing. I thought it was like a thing that you play. All right. Well, I don't know. I'm not going to find it now.
I can report on the weather here. We're at about 0C. Bright sun. We had a snowstorm move through last night, but bright, sunny, and a little bit of snow on the ground.
Yes, Calliope herself. It's my name. Calliope is a goddess of inspiration, one of the muses, and that's why I keep the full name and don't shorten it. Too much history.
Nice.
You know, there's a lot of pressure on you, Calliope, because you have to be an inspiration to people now, right? You have a name like that, you got to inspire people.
I think it's working.
Oh, yes. It's also an organ, the Calliope whistle. Right.
Oh, that's what you're thinking of.
That's what I'm thinking of. Exactly. Yeah. I didn't think of the goddess at all. Well, thanks, everybody, for coming here today. We promise a informative and perhaps entertaining. I would say this is going to be a shocking episode, Nels. What do you think?
A shocking means to move genes. Yeah.
Shocking means to move genes. Wow. That's great. Hey, that's the title. A shocking means. Nels is very clever with the titles. He rivals. Alan Dove on TWiEVO.
I concede. Alan, I think, is the true sort of master of teasing out good names for podcast episodes. But because I pick the papers on some occasions, I can kind of merge the title with the paper. And because we were kind of scrambling a little bit to get in 3 podcasts in 1 month, I picked a paper that might be a little off the beaten path here a little bit, but I think, and there's, let's be honest, this probably isn't going to be the most groundbreaking work, Vincent, that you and I have kind of put forward, lifted up,
Yes,
put forward, and sort of chewed on here with podcast peer review.
it's true.
But it's pretty fun. And honestly, sometimes in these like kind of surprising spaces, I think actually it can sort of inspire in ways that might not be expected. And so that's kind of the spirit that we're bringing this paper forward with. And so the paper, let me get to the title here. It's Electric Organ Discharge from Electric Eel facilitates.
There we
Oh,
go.
great. Yep. Facilitates DNA transformation into teleost larvae in laboratory conditions. Published, I think, in December 2023, a few months back in the journal PeerJ. This is a group coming out of two universities in Japan, Nagoya and Kyoto universities. First author is Shintaro Sakaki, and the last author is Atsuo Ieda. And so why would we put this forward? Pretty wild. So we'll try to decipher the title here. And why would we put this forward as a topic for TWiEVO, This Week in
Nels,
Evolution?
before you talk about that, tell us about PeerJ. What is that?
Oh, yeah. I actually, I don't know too much about it. It's open access. Do you know,
Right.
are you familiar with it, Vincent?
I just
PeerJ?
know it's open access, right? And it's another one like bioRxiv and so forth. I don't tend to look
Yeah.
at papers here all that much. I don't know why.
Correct. And so, yeah, so there's a number of journals, I think, that are kind of on the forefront of experimenting with how we, as scientists, share data with the community. And so probably, one of the most talked about examples of this is a journal called eLife that has really been playing around with the publication model. And so, the traditional way this works is that as an author, you spend years doing research usually, or invest a significant amount of time and energy into that, and then you reach out to a journal, an editor. You go to the website and reach out to a journal, kind of based on where you think it might be a good match, and you're making contact with an editor, either a professional editor or a sort of pseudo volunteer editor, an academic editor, someone who's running a lab or has a research group, but then also is appointed onto an editorial board. And then if the editor in the traditional model thinks it's an interesting paper, interesting manuscript, then they'll launch peer review, meaning they'll send it out. They'll try to identify peer reviewers that will research or will read the paper, give critical feedback. Should it be published? Does it need modifications? Are there weaknesses hiding here? Does the sort of evidence match the claims is a big part of that. And then if that's the case, then, the paper might go through a revision or two and they get published. And that process, kind of time honored, but can also be sort of time intensive. It can take years in some cases for a paper to find the light of day as it sort of moves through that. And so eLife, other journals, I think PeerJ falls into this category, really just put the work out first. Peer review is kind of happening in a very open or transparent way in a lot of these cases. And the author has a little bit more discretion on, what's the sort of publication of record, so forth. And PeerJ, I believe, kind of falls into that open access category where they're really kind of going for transparency and openness. A little bit off the beaten track. The way I came across this paper was it showed up in my Twitter feed. I think someone just was kind of, caught by how sort of exotic the topic was here. And so let's talk about that. So
I made a mistake. Yeah. Let me correct it. I said this was a preprint. It's not. It's a final reviewed stuff. It's not like bioRxiv at all. It's just a different journal. And
correct,
it has a very wide coverage. It covers a lot of different sciences.
Mm-hmm. Yep. PeerJ. And so the work here. So actually, let's even step back and sort of why would we kind of grab something that sounds a little bit obscure here? Electric organ discharge from an electric eel. That's sort of familiar, but
let's talk about DNA transformation. That's cool.
Yeah, no, exactly. And that's where the authors kind of come from and kind of caught my attention as well. So before we get there though, Vincent, let's talk about how in evolution, how we usually think about how genes move from, let's say, individual to individual in a population. And that's generally by what we call vertical transmission. So the parents might get together. There might be some recombination happening, and then the genes move from parent to offspring or mother to child, if it's a mammal, for example. And we call that vertical transmission. It kind of goes vertically from generation to generation. But as people are sequencing, as the field, the community, and broadly in science are sequencing more genomes, what's becoming clear is that the rules of vertical transmission are oftentimes, not often, but on occasion, are broken. And we often call this horizontal gene transfer. And so here, instead of from parent to offspring, the genes are moving from individual to individual, and even can move between species, and even can move between kingdoms of life in some of the more dramatic cases. And so I think this raises, the obvious question: how does this work? How do genes move horizontally between different species? And we've got some clues in some cases. There's some really clear mechanisms in bacteria and the viruses of bacteria. That's sort of a well-described mechanism for genes sometimes hitching a ride in a virus genome or associated with a virus and then moving to another individual in the population. But for, animals with backbones, fish, for example, we see some hints of this, but the mechanism or the means by which this happens is less clear. Mm-hmm. And so that gets us kind of to maybe the starting line of this paper. And I think the authors almost did a reverse of how we usually think about evolutionary biology, or even mechanisms of biology. So usually what we do as scientists, as curious people, is we look at observations in the wild, or bring animals from the wild into the laboratory, set up what's sometimes called model systems or research organisms in the lab, and then try to get them to behave the way they might actually behave in the wild, but then do experiments to sort of understand the biology behind the behavior, the phenotypes, the sort of different characteristics of those living systems. In this case, I think what's really fun is the authors sort of said, well, let's see what the scientists do in the molecular biology laboratory, and then ask in reverse, does that apply to the natural setting? And so for, 50 plus years, scientists have been moving electric currents through embryos or cells, a process called electroporation, to move DNA genes across cell membranes. And the way electroporation works is that it takes advantage of the fact that DNA as a chemical is negatively charged. And so if you push electric current, you have DNA and current together, this can move DNA. So a really common technique in the laboratory, gel electrophoresis, also takes advantage of this property to separate DNA molecules by size. You can also physically move DNA with electric current across cell membranes using that same principle. And so the authors, I think, were kind of wondering, well, okay, if scientists do this, does this happen sort of in the wild? Or is our behavior here moving DNA? Could there be a sort of a natural correlate of that? And they're not the first people to totally think about this. I think there's been some suggestion actually that lightning strikes could be a natural source of electricity, that if there is, somehow DNA, like let's say a lightning strikes a mud puddle or something like that, or a pond, that blast of electric current, if there's free DNA, could that actually move it across cell membranes to transform some of the cells there, actually, so they would take on nucleic acids? This would be horizontal gene transfer, or an example of that. Pretty limited evidence, to be generous, that this is happening at an appreciable rate. And certainly, I don't think anyone has sort of seen it in action.
Now, in nature.
Yeah.
So viruses can move nucleic acids from cell to cell, but also plasmids have ways of getting into bacteria, right, through pili. So they can come out of the bacteria as they lyse and move into other bacteria. But eukaryotic cells, they can take up DNA without any special conditions. Like amoeba, which are highly phagocytic, probably take up a lot of DNA, right? And I think that they're kind of garbage disposals in a way.
Yes, in a good way, in a genetically interesting way. Yes,
macrophage. Yep. But none of this is germline stuff, right? If it's persist.
Yeah, agreed. If it's a single cell organism, it's germline. Yeah. And the authors kind of hint at that. They're talking about E. coli. Right. But I think what you're getting at is an important, or several important barriers here. So if a macrophage or an amoeba takes up some DNA, which is happening constantly, there's still at least 2 cell membranes that DNA has to cross before it gets digested for it to actually have the ability to be inherited, right? So it has to get across the phagosome membrane. So the phagosome, that's this compartment that forms that is sort of tasting the environment and then usually leads to cellular digestion. But there's all kinds of cases of viruses or bacteria that can kind of get out of the phagosome. But then in the eukaryotic cells, as you're pointing out, Vincent, right, there's a second barrier, which is the nuclear membrane, where DNA lives, where the genome is housed, where you find all of the chromosomes. And so if for our sort of environmental DNA to undergo this horizontal gene transfer, it has to get across, in that case, at least those 2 membranes. And so that electricity can do the job.
So let's go back. First person to publish this was in 1944. Avery, McLeod, and McCarthy. They put DNA into bacteria because at that time people didn't believe that DNA was genetic material. They thought proteins, but they thought otherwise, and they said, let's check it out. And they put DNA from one E. coli into another, or one, not E. coli, but streptococcus, I believe, and transformed the morphology, and that really established that DNA was the genetic material. But they they modified chemically the bacteria, and that's what I used to do in the lab all the time. I would treat E. coli with a solution of calcium, which made it more, we say, make competent cells, which means they're competent to take up DNA.
That's right.
And then the electroporation thing came along. I never used it. I thought it was just this newfangled thing I didn't need. I could get DNA into cells. I just push it in, no problem, with some calcium. And but we had the electroporation apparatus. You probably have them in your lab too, right now.
We do both. We make cells competent, just like you're describing with calcium chloride.
Yep.
We also have an electroporator. For my PhD, I spent countless hours with both with the electroporator, but also the gene gun. And by this, I mean, like literally, we would take, so we take a membrane, put gold particles, coat it with DNA, and then shoot it into the cells.
Right.
This is a eukaryotic cell, and it works. So there's scientists have dreamed up all of these ways to get DNA material into the membranes. And then if we move from E. coli, like you're saying, I mean, this is you're describing kind of the foundational work at the dawn of molecular biology, before the structure of DNA was known. I mean, I love this. It's so exciting. But then, so scientists have been doing this for a while. Electroporation turns out to be one. I think you're, in most cases, chemical, certainly for single cells, and for bacteria, chemical transformation is sort of the way to go. But for more complicated scenarios, electricity can help.
To electroporation, you don't have to prepare the bacteria in any way. You just mix them with DNA and a little cell that has electrodes in it, and you apply a current. Whereas to make them competent with calcium chloride, you have to prepare them ahead of time. It's an hour or two procedure. It's not very hard. But then you freeze them down, and you go and you pull them out, and everybody runs out. Can I borrow your competent cells? You give them some, and then you don't have any. That's right. Yeah, it's a chore.
Thing. It's a chore in the laboratory. People, they have their turn to make competent cells. That kind of wastes and uses up an afternoon of time. It's sort of a chore.
Do you buy them? I bet you buy them too, because
it depends. We still make them ourselves. There are different situations where having that control is useful. We also have an electroporator.
To buy the bought cells, you can have very, very high competence. So competence is measured by the colonies you get with a microgram or a nanogram of plasmid, and you use antibiotic resistant to measure the colonies. So in other words, you take a bacterium that has no antibiotic resistance plasmid, and then you introduce a plasmid with antibiotic resistance and antibiotic, and then you plate in the presence of an antibiotic, and you count your colonies. And a really efficient bacterium, I don't know, we used to be happy at 1 million colonies per nanogram. Am I thinking correctly?
Yeah, you are, and usually you don't need 1 million, so you can get away with a lot lower.
Of course. But if you want to make libraries and stuff, if you want to make really good libraries,
that's right.
You do that,
Yeah. So there you go. So the molecular biologists have been doing this for a while. The authors, then, kind of getting to the punchline of this work, say, while electric organ discharge is primarily associated with behaviors related to sensing, predation, and defense. It may incidentally serve as a possible mechanism for gene transfer in the natural environment. So the question then is, can electric eels step in for your electroporation
It's amazing. It's
device?
amazing.
And you
Nels,
mentioned,
the whole thing about these eels making electricity is so
yeah,
cool. I haven't thought about this in a long time. They make
yeah,
like up to 186 volts.
I know. Yeah. And I think,
More than,
and so, yes, they make a ton of, so they're powerful. The electric current is strong enough. You could imagine it's in the range that you could
well, current
imagine.
is not voltage, right? Current
Oh,
is amperage, right?
yeah. My electricity, knowledge of electricity and the terms around it is pretty primitive. But
It's voltage,
yes,
yeah.
but up to voltages can reach up to 860 volts, they claim in the
Yeah.
abstract of the paper. And even more than that, it has, so like, it pulses, and that's very similar to electroporation as well. So the techniques that have been used, and you can, depending on the protocol you're using, the cell type or the animal you're trying to transform with DNA, if you're using electroporation, there are all these different programs, different waveforms, different sort of pulses that we sort of artificially provide. Electric eels do this kind of as part of their own natural, as sensing, predation, defense, but has in some cases waveforms. They're bell-shaped instead of square, et cetera. There are some small differences, but pulses in ways that is plausible that it might work. And so that kind of sets the table here. So a pretty simple scheme, a little bit artificial. So the authors definitely admit that this is like,
That's so funny.
kind of
It's really a gas. I got to
it's
tell
great.
you.
Yeah, I love it. So it's, so they got a fish tank, bought an electric eel from a aquarium store or something like that, or a aquatic supplier.
I got to show you this picture here.
Yeah, great. Put it up. They put the electric eel into the tank, and so then there it is. There's a little, both a cartoon there at the top and the actual photographs at the bottom there. So the eel, I mean, it's kind of taking up the whole tank. It's probably not super comfortable there. And then on the right panel, panel B, you can kind of see here's the work of the, of the setup of the system. Is they've got some zebrafish. So it looks like there's a orange fish. You can see that's a goldfish. That's actually bait. That's a meal for the electric eel, because when it eats, when it makes contact with I think it's an anesthetized goldfish, that will provoke the electric current. And so you've got a negative current on the left, positive current on the right. It's grounded. As I mentioned, DNA is negative. So as an electric current shows up, it'll go towards the positive pole. And then you mentioned you called it a cell, but it's also called a cuvette, which is, and so you can see some tiny fish in there. Those are zebrafish larvae. It turned out, I think they did through some trial and error, it made sense to use 7-day-old embryos. That's kind of a common time spot in developmental biology, where the fish is sort of moved into an independent life state. It's kind of stable and robust, but also still very early in the embryogenesis. And the cuvette has, so this is like, there's that's a very, I think, important question here. If we do want to sort of launch the hypothesis that something like this could be happening actually, in nature, in aquatic settings where eels are swimming around and eating, or, discharging electric pulses, there's not going to be obviously a lab cuvette with 2 electrodes to help facilitate the movement of the DNA here. So in that cuvette is DNA and our embryos, and then the electric pulse is provided by the eel. And the notion is that movement of DNA could actually move through the cell membranes and into the fish. The DNA they chose is a plasmid. It's a circular piece of DNA. That's in panel C. Really going into the weeds here, it's 8,888 base pairs. Each base pair is a letter, A, T, C, or G. And it encodes for green fluorescent protein, or GFP. And so it's kind of a clever idea. So if it's under a promoter, beta-actin promoter, which is strong, means it will strongly produce copies of the gene GFP into RNA that will get translated into protein. I'll have this jellyfish protein. So that's where GFP is from originally. It was isolated from the genomes of jellyfish that glow green. And now you're a zebrafish, or whatever animal that you put GFP into will glow green. And so that's kind of the whole, the take-home message or the sort of impact of the paper is shown in panel D there. So you've got these two embryos. One was put into that whole setup without that plasmid, without GFP, and the other was with. And then the electric eel shocked, and then you put under a fluorescence microscope those embryos and ask, is there signs that the GFP is produced in any of those cells? That would mean that it worked, that the plasmid DNA moved across or from the water in the cuvette, the buffer in there, and then it moved into the fish. The plasmid is now maintained in the zebrafish cell, and the gene is decoded. It's translated into protein that glows under the fluorescent light green, and you can see in that lower right panel of D that there's more GFP signal in the one that had the plasmid versus the one that didn't.
You can even see its little eyes there and its mouth that got a lot of GFP.
That's right. There's some GFP. The authors kind of grapple with this. There's also the case that you can have autofluorescence at these times, and so you can see that a little bit in the top one. There's you might ask, why is there a little bit of green? That's probably right around that's the GI tract. The embryo started eating a little bit, and some of the stuff it's eating might be a little bit green. It's probably not GFP being expressed. Autofluorescence is very common, and just through the process of digestion, it's really rare, although not impossible. But certainly, kind of, in this experimental scheme, they wouldn't expect to see GFP being digested and then reporting it. Most of that DNA will get eaten up and actually used for nutrition. So there's something different about that electric current going on there. So then that's kind of it, Vincent. It's sort of, I think that I don't think there's much more to say in terms of that's kind of the one experiment they did, and then they sort of quantify it. And so, but I think what it raises then is the question of, okay, so now let's go back to real, to a pond environment, not an experimental setup. And is there a situation where this might be happening? And so the authors, I'm going to return to some of the authors' words here in the discussion, because I think they're trying to put connect the dots for us a little bit. And so they're pointing to some work that's been done in the Amazon River, where you can find some electric eel species. And they say, so their findings that you can get the electric eel to, in this very, experimentally kind of contrived system or setup system, but they say the findings support the notion that the hydrosphere of the Amazon River contains the necessary components for natural electroporation, including the electric eel as a power source. And live fish, not zebrafish, zebrafish are from Southeast Asia, whereas the Amazon is a whole different set of teleost fish or bony fish. The live fish has recipients and eDNA or environmental DNA as a source of genetic factors. And so there are a number of studies, some on microbes or some on viruses, where people will take water from Amazon River, from the ocean, from wherever, and do metagenomics, try to sequence all of the DNA that you find in water. And it's pretty rich with DNA, both which can be associated with things like virus particles, packaged single cell critters, both bacteria and eukaryotes. But also you start to see, even in these samples, fish DNA, for example, which could be the process of, cells sloughing off, the digestive process. There's a lot of genetic material just sort of floating around, or eDNA, in this case, environmental DNA. And then the authors say, we hypothesize that horizontal gene transfer may occur among organisms, including fish, in the habitat surrounding electric eels.
Yeah, this eDNA is everywhere. It's actually, people use it to find out what kind of fish are living in a stream, for example. They just take the DNA and they sequence it, and they say, okay, so and so is living here.
That's right. Yep.
But,
yeah, and there's these. Yep.
The thing is, Nels, the zebrafish, it looks like it was mostly surface cells that got the DNA, right?
Correct. Yeah, and I think what you're driving towards is a really important, not just a detail here, but an important feature for this to kind of make sense from an evolutionary standpoint, which is the issue of heritability. And so, in addition to getting into those sort of surface cells where they see some evidence, so that wouldn't be inherited, right? So it's not clear,
right,
whether there's two steps here. or maybe 3 steps. So you have to get across the cell membrane. It looks like that happened in a few cases. The next step is it needs to get integrated into the genome.
Mm-hmm.
That's not clear. They didn't test that or look for that, and that's okay. I mean, I think that was sort of beyond the scope of what they were going after here. But then, from an evolutionary standpoint, not only does it need to integrate into the chromosomes, but if it's a zebrafish, it needs to get into the germ cells, the ovaries, or the testes,
Mm-hmm.
and then that new gene, as when it's integrated, needs to be encoded in those genomes that will go on and be passed to the offspring. Then you can imagine that there's sort of a genetic mechanism in place.
And you could test that, right? You could easily do that in the lab. You take some of these fish,
Yeah.
you grow them up, you mate them, and see if they transfer the plasmid, for example, right?
Yep, absolutely. So, it also just sort of points to, so I think, for me at least, this is plausible. I mean, I think it's super low probability that this is happening in a lot of vertebrate animals. The amount of horizontal gene transfer that we've documented or observed is relatively modest too.
Mm.
So these are super rare events, but you could imagine like a jackpot scenario if this worked. And so, could it be an electric eels doing this? Sure. Or what is a more likely sort of source of this? And I think, for my money, my bets are on the viruses as sort of the mechanism, the means to move genes. And there's some evidence of this, other published papers. We have hinted at it from time to time on some TWiEVO episodes. But think about it. The viruses are just sort of, they're the masters of the mechanisms at play here. So not only do viruses contain genetic material, they package it really efficiently and tinker with it. There's high mutation rates, the ability to gain genes from hosts for many viruses, even diverse classes of them. They also know how to get across membranes. So they, as part of the replication cycle, sort of by definition, they need to get into the cell, move around, in some cases in many cases, get into the nucleus. So they've now crossed 2 membranes, replicate, and then get back out. So they've got all of the right tools to do what needs to be done when it comes to the horizontal transfer of genes. And as you're pointing out, this has been, long known for the viruses the phage that infect bacteria. And I think more recently, including some of the work that we do in my own group, thinking about how viruses might be moving these genes as well. But I think it's really important and fun to think about like all of the other mechanisms that are possible. It's sort of like relate them to each other. That could multiple things be at play. Are the viruses sort of increasing the level of eDNA, environmental DNA? How does it get taken up? What are the sources? What are the collisions or interactions in these ecosystems? And then this sort of really fun notion that, either if there's a lightning strike on that lake or a electric eel hanging out feeding nearby, that this could actually facilitate or increase the probabilities of these really rare events happening.
I think that's the key, increase the probabilities, because I think DNA is taken up a lot because it's everywhere. Organisms take it up, and it's just a matter of whether it gets in the nucleus. I mean, there is you can imagine that DNA would just be taken up passively by endocytosis, which happens constitutively, and then it would get in the cell. Getting in the nucleus is, Nels, don't you need a protein to get in the nucleus through the pore, because DNA and RNA won't get in on its own? There's no import signal, right?
Yeah, that's right. So so a lot of proteins encode what's called a nuclear localization signal, or NLS, that helps facilitate. When we think about just the eukaryotic sort of cell structure, having that extra membrane could be a really effective way to protect the genome, to keep other genes out. And so then that's, when you set up a barrier like that, you see other mechanisms. So the nuclear pores are these complicated protein machines where proteins are constantly, being shuttled in and out in a directed way using these nuclear localization signals, nuclear export signals. So kind of traffic in both directions. And then of course, immediately, what do viruses or other sort of replicative elements, or to transposons, what do? They figure out ways across those borders, so to speak, and then facilitate the movement of nucleic acids. This is also happening just as in the common course, right? So just when we think about the translation of proteins that get into the cytoplasm. The organization of the ribosomes, the protein machines that take messenger RNA, translate that into proteins. You have to move that nucleic acid, those nucleic acids, the messenger RNAs, across the in some cases the nuclear membrane as well. So there's all kinds of mechanisms in place, and then the cheaters show up and try to take advantage of the resources or the sort of genetic conduits in different interesting ways. Yeah.
So do we know what proteins bind DNA in the cytosol to help get them into the nucleus? I'm not aware of any of that.
I think the answer is yes. I'm a little rusty on this, and so I'm going to take it easy on speculation here at the moment.
Okay.
But yeah, there's all kinds of
something is binding the DNA, right?
Absolutely. And so the example that comes to mind immediately is retrotransposons. So the line 1 elements, for example. So it's relatively simple. It's a copy and pasting mechanism, so-called selfish genes. In our genomes, the human genome is chock full of these things. About a quarter of our coding capacity is devoted to line 1 elements, something that isn't sort of a core genetic function that we would think about it that way. Contributes to the diversity of genomes in interesting ways, and that's an area of active research. But the mechanics of this is, so the retro elements, or the retrotransposons, encode a couple of genes, ORF1 and 2, that then become proteins. They associate with RNA out in the cytoplasm of the cell, and then bring that back into the nucleus to copy and paste themselves. The RNA is a copy of themselves, basically, or a copy of the transposon. And in that mechanism, they actually do go over and across the nuclear membrane and back in the replication cycle. Yeah.
So when I was a postdoc, Nels, I took a plasmid, a DNA encoding poliovirus genome, right? Now, this is an interesting point. To get DNA into eukaryotic cells nowadays, back then we used a technique called calcium phosphate coprecipitation, and we would mix the DNA with some calcium and phosphate solution. It would precipitate out, and then you would apply the precipitate to cells, which would then take it up, because cells are always taking up stuff from the external environment. You didn't have to do anything except do the calcium phosphate. I have to say that when you made the precipitate, you had to look at it. It had to be like a fine, hazy thing. If it were clumpy or nothing, you couldn't use it. You'd throw it out and start again. It was like black magic. It was just ridiculous. But then the DNA would get into cells, and then it would get in the nucleus, because that's where transcription happens. And so some fraction of the DNA gets in the nucleus in transcription. Then you, in our case, that made polio RNA and started an infection. But you can also do that with GFP or whatever. So the DNA gets in the nucleus, which to me is weird, because the cell shouldn't want external DNA coming in. That's probably not a good idea, right?
Well, so interesting question. And again, I think, from first principles, if we put that into an evolutionary context, so what is DNA, sort of the inclusion of DNA or new DNA, sampling DNA, what does that do? And I think, we've talked about this. It can be like, it's a mutagenic process. If you change your genome, by definition, you're mutating it. And like in most cases, that's a problem. That's deleterious. Kind of our notion of mutations being bad kind of builds out of that, and is true. So like in the majority of cases, most mutations are detrimental. A lot of them are neutral. They don't really matter for a number of reasons. And then there are rare ones that are advantageous. And so that kind of sets the stage for thinking about, if there's some level of sampling of horizontal gene transfer, can this be kind of a genetic or a evolutionary adaptation in some rare cases? And then can that sort of get cooked in to a bigger scheme where if you had perfect replication, evolution would sort of be at a standstill. And then, if there's other things that are diversifying or mutating around you in rare cases in beneficial ways, then as things collide and different, animals or microbes are vying for energy, very quickly, advantages and disadvantages start to appear around the mutation rate, right? Yeah.
So mutation is maintained because it gives rise to diversity. And same thing with taking up DNA, right? It gives rise to diversity, so it's not going to be negated in any way. So Although when DNA gets in the nucleus, there are some responses. Like
Oh yeah, yeah.
if
Immune
single-stranded
responses, right?
if single-stranded DNA gets in, it is repaired to make it double-stranded because the cell thinks it's broken DNA. And then
Yes.
once it's double-stranded, it gets coated in nucleosomes and silenced. That's the way you silence
Yeah.
DNA
Yeah.
transcriptionally, is to coat it in proteins like histones. It will become transcriptionally silent. And then if you're a virus, you need to figure out a way to get around it. And they do, of course.
Yeah,
So
that's right. And this, I mean, I love this because it kind of echoes on from the paper that we've been describing, which is that becomes so when you get foreign DNA into a cell, like you're saying, that sets off all these alarm bells for the cellular immune system,
yeah,
and either you silence that DNA, which is, if you're a molecular biologist, you want that DNA on, whether it's, in this case it's just GFP reporting, but you might want to express a gene to test an idea, et cetera, and so or disrupt a gene or whatever you're doing. But if immediately gets turned off, or the cell commits suicide because it thinks it's infected with a virus or something like that, and that's part of a bigger immune response, then the very thing you want to get for your experiment is gone. And so what's happened over time is that, scientists have done these experiments. They've kind of found cell lines where the transformation efficiency is higher, not necessarily because you can get more DNA across those membranes or get it in position, but because there's deletions of immune genes, so that even though there's foreign DNA there, it doesn't set off those alarm bells. The cell population is just more kind of quiescent or willing to accept this, the transformed DNA that the experimentalist is providing. And so that gives us clues back about, okay, well, what are the key immune defenses in these cases if you start kind of taking them away in order to get higher transformation?
So I want to get more into the weeds here. So, obviously in the zebrafish experiment here, some of that DNA gets in the nucleus and it's expressed, so it's not all silent. So there's some fraction. I don't know how much, but if you remember, I think this was a thing. You add a histone deacetylase inhibitor with your DNA, and you get even more expression. Is that correct?
I think that's right. Depends on the system and the setup, et cetera. But as a general rule, yeah. And so and actually, there's a perfect example to me of like how just messing around in the laboratory with one goal, which is to get DNA into a cell and then the genes expressed, can lead to all this insight into the biology behind either how the immune system senses this, how genes are regulated, and then kind of these knock-on effects years down the road and sometimes decades down the road, where as we understand this with the systems that we started with, we might apply that knowledge or that new information to intervene in ways when we want to control the expression of genes for sort of therapeutic reasons. So this is just one of those, kind of another example of how kind of almost stumbling around or literally going to the aquarium store and buying an electric eel and putting that together with zebrafish in this contrived setting can sort of advance just the scientific conversation, or just our scientific kind of brains kind of stumble forward. And then you end up with, potentially all these cool ideas that wouldn't happen unless you were just sort of messing around. Yeah.
Be curious. So let me explain the histone deacetylases. So DNA is wrapped around histones. They're called nucleosomes. Imagine them as a little, I don't know, a cylinder, a flat cylinder. The DNA is wrapped around it, and the proteins that make up the flat cylinder can be modified by methylation or acetylation. If they're acetylated, it makes the DNA wrap more loosely around this core, and it's easier transcribed. If you take off the acetyl group, the chromatin becomes compacted, and it shuts off transcription. It's a good epigenetic way to regulate gene expression. And so you can put in inhibitors of there are enzymes in the cell, histone acetylases and histone deacetylases, that can go and regulate gene expression. And a deacetylase will take off the acetyl group and shut down transcription. So if you use an inhibitor of histone deacetylase, you can get better expression of your gene when you put the DNA into cells.
Yeah. And this is important. So both for like fundamental gene regulation, but also like the way as you're describing this, Vincent, what it reminds me immediately of is how virologists are trying to intervene for things like retroviruses, like HIV. So one of so why haven't we cured HIV, right? Many reasons. It's really hard to get an effective vaccine, et cetera. But even with an effective vaccine, part of the challenge with HIV is that it reverse transcribes itself. It's a virus that has figured out how to not only get into the nucleus, but then take that message and integrate it directly into the DNA, flashing red light of horizontal gene transfer here. But what happens is oftentimes in those blood cells that have an integrated copy of HIV, it becomes part of, and it's been proposed in some cases, part of the immune defense is to wrap it around histones with those acetylation marks, those patterns. And then what happens is it becomes quiet or latent in the cell. It doesn't express any of those genes. It just hides out there. And so why is that a problem? Well, if the virus can kind of hide out in the genome, you treat it with, a triple cocktail therapy, these, great interventions, medical interventions that we've come up with. But, even it just sort of, it's hiding out, it subsists. And then right when you either let your guard down, you don't take the drugs, or, the immune system becomes compromised or changes a little bit, all of a sudden, or those acetylation marks change just enough, unwraps from the histones and becomes transcriptionally active again. And so this is sort of a major area of active HIV research right now, is to try to sort of shock the cellular system to perk up that virus, to kind of get it out of hiding, and then hit it with drugs. And so people are adding these compounds that will influence the acetylation patterns, which will then influence how the DNA is in chromatin or in heterochromatin, where it's quiet, or chromatin, euchromatin, where it's more open and vulnerable to being transcribed. And so, and then it sort of those infected cells reveal themselves as being infected, and then other immune responses or treatments can kind of come in. And so that's sort of the hope for, advancing more effective therapies, is to actually get the virus out of hiding so that it can't just persist there over months or years or decades in the case of HIV.
Let's see here. We have some comments. Let's highlight here. Sean says Nagoya is a sweet town. That's one of the universities. Yeah, I've never been to Nagoya, but the other town is Kyoto. I've been there. It's very nice. Yeah. Kyoto's lovely. Kyoto is one of the cities that was not bombed during World War II, and so all the old buildings are still there. It's very nice if you have a chance to meet it. Sean says, wow, electroporate using lightning. I think the DNA would be denatured.
That's for sure. And actually, I'm glad you're bringing this up, Sean, because the authors here point out that, so they actually were trying this. Why did they use 7-day embryos? They actually tried fertilized eggs and then earlier embryos, but the electric pulses from the electric eel in that case was too strong. It killed them all, or killed most of them. And so they had to kind of find the sweet spot. I think for electroporation by lightning, you're right. If it's a direct hit, all bets are off. But the question becomes, what's the sort of electric blast zone, so to speak? And then are you kind of at the margins where it's just that sort of Goldilocks position of getting enough electric current to move nucleic acids across membranes without being so disruptive that it kills the animal? That happens in electroporation too. Like if you're electroporating, even if you're electroporating E. coli, for example, you'll tolerate something like even half the cells dying in some cases in order to get, the other half to have a shot to get. And so it's sort of a balancing act, even in the laboratory as well.
Michael Schmidt's here. Hello, Michael. Volts versus amps. Yeah. Like volts is the differential between 2 points. Amps is the current, the flow of the electrons. And if you multiply volts times amps, you get watts.
This week in electricity, a new podcast is born.
I probably got it all wrong. Nice to see you, Michael.
Yeah, great to have you here.
Vanity Nutritionist joined us. Thank you for coming.
Great.
Moderating. And I also noticed that Steph is here. Yes, here's Steph. Good morning. Hello, Steph. Fantastic. Welcome. 2 moderators. Thank you for joining us.
Yeah, appreciate it.
Let's see. Yes, this is good. Les says on December 10th, 2008, Osamu Shimomura, Martin Chalfie, and Roger Sien were awarded the Nobel Prize in Chemistry for the discovery and development of green fluorescent protein. Originally came from a jellyfish. Martin Chalfie is a professor here at Columbia University.
That's right.
And so if you've ever been, say, in a bay, a saltwater bay where there are jellyfish, you go out on a dock and you take a net and you just move the water, you'll see the jellyfish emit light, right, as you do that. It's so cool because
Hmm.
they're full. The water's full of them. And all you have to do is disturb them and they emit their light.
Well, yeah, there's all kinds of sources of light and critters in the ocean. So the dinoflagellates that there's some great examples of bays where you can, like, even if you're like rowing or kayaking, every paddle, like, it lights up like a, and then yeah, the jellyfish. It's not just GFP. There's now
Yeah.
red fluorescent. There's sort of a whole rainbow of this that all came out of
Yeah.
some of this original work.
Yep.
From this trio. And so why would fluorescent protein from a jellyfish win the Nobel Prize in medicine, right? Physiology or medicine. And so this has been for basic science, the ability to see, in this case, to have a gene, that kind of direct connection between a gene, the protein, and to see this in living cells in an animal has been super powerful, not just for,
Yeah.
kind of basic, like, it's been the creativity around using GFP to kind of see the proteins for your eyes in living cells in
Living cells.
tissues,
That's the key.
right?
Yes. You don't
That's
have
the
to
key.
fix
So
them.
that's it. So we've had, there's been hundreds of years of microscopy with fixed sections, and a lot of work into the technology of how do you fix something to try to recreate, something that approximates the living state. But these are, by definition, snapshots of a biological process. You have to kill something in order to see it. And for most of microscopy, the revolution here with GFP is that now you can do this all in live cells, live animals, and see biology kind of as a movie instead of a snapshot. And it's just been, totally inspiring and led to breakthrough after breakthrough.
So as Nell said, you can find different colors in different jellyfish, but you can also modify
Mm-hmm.
the protein and get different colors as well. You can have over a dozen different colors like yellow, cyan. There's one red. There's one called tomato, slightly different
Yeah,
from red. And
that's
you
right.
can buy fish that have incorporated these things into them so they glow different colors as well.
Mm-hmm.
Do you have glowing zebrafish in your lab now?
We do. We can't get enough of it. So our our glowing zebrafish we threw in our own little trick here. Is we hooked up GFP, not to so in this paper that we're talking about today, they hooked up to an actin promoter. So as soon as it gets in that cell, it's turned on. We hooked up GFP to an immune responsive promoter. So when there's immune signaling happening, that turns on GFP. When we launched that transgenic fish, all of a sudden, we didn't use an electric eel to transform the fish. We used some other techniques. But when we when we made those transgenic fish, immediately they started turning green. Like the intestinal tract, we had to compare this to autofluorescence. They started turning green. Some of the other cells, this was the immune response happening. And then we took those fish that turned green, compared them to the ones that didn't, sequenced a bunch of RNA. We discovered the first natural virus of zebrafish, a picornavirus that replicates in the GI tract, in
That's
the
very cool.
intestine of zebrafish. So GFP gave us that. The immune response is usually invisible.
Yeah.
GFP gave us that made it visualized an immune response in the cells that were triggering or responding to immune signals, and then we used that to do comparisons that allowed us to discover a virus for the first time.
And a number of years ago, Lynn Enquist, who was a virology professor at Princeton, did a very cool experiment where
Yeah.
he incorporated different color fluorescent protein genes into herpes virus, a herpes virus, and he made, over a dozen different colors. And then he put them all in cells at the same time to find out how many would get into a single cell. And no matter how many you added, the maximum you could get was 7. And you could see 7 different colors
Wow.
in the cell, and that's it. No more. So if you put 12 in, you would not get 12 different colors. You would just get 7. So there's some kind of exclusion going on there.
Yeah, that's cool. The neuroscientists have taken advantage of all those colors. The rainbow, both in mice and in fish, where you have different cells, in that case, randomly expressing some subset of them, but then you get kind of variations on the colors, and you can really discern one cell boundary from the other, and sort of the rainbow of fluorescent colors here. Yeah, really fun genetic science.
Rob says Mary Shelley would be impressed. Yes, indeed. Any other questions, folks? If not, we're going to wrap this up. Looks like no. Oh, we have to do some picks, Nels. I forgot about that.
Yeah,
Let's do that.
let's do that. So our science picks of the week. I'll get started. Um, so I kind of saw. I took a peek at your pick a little bit ahead of time, which is a nice art theme here. I tried to kind of piggyback onto that, and so my pick of the week is a quilt. It's called The Jump. It's by a quilter, Cristina Bono, I think is from Spain. And I've got a link to this. So it's in the quilt itself. It was inspired. Here we go. It's inspired by a Russian author and artist, and kind of on the theme or aquatic theme. So instead of an electric eel, we have a girl in a striped red bathing suit jumping into a pond and all of the fish scattering around there. Incredible detail. So check out the link when we put this up at the TWiEVO website, and you can really kind of scroll through and see the detail of this quilt. Also, the original print is really impressive too. Yeah, there's a close up. Look at that. The applique and the artistry there. Yeah. And I think for me, it was a little bit echo of, jumping into the pond and kind of disrupting the waters around or the critters around to kind of build off of the topic of our
It's cool.
The topic of our show today, The Jump.
So this is inspired by, I see. Oh, I see. It's Sveta Dorosheva. Yeah, it's like you said here. That's very cool.
It's the author who did the original print that quilt was based off of. And this is the first quilt that we've had on TWiEVO. It took us 99 episodes to get to a quilt, but that's it was really science pick of the week. But I think I like this idea of kind of maybe an art pick of the week that connects some way to the science that we're talking about, which leads us, Vincent, to your pick of the week. What do you have for us?
So this was sent to me by Mark Martin.
Mm-hmm.
And this is the main part of this is by a beader, Gwen. Mm-hmm. She has a website her name is Gwen Fisher, and her site is called Gwen Beads. So she makes things out of beads, and she made these two phages.
That's cool.
Which are all made out of beads, right?
Yeah, that's
neat. They're beautiful, right? You have the icosahedral head, as you can see here, and the tail and the tail fibers, and they're all beads, which I think is just very cool. Look at that.
Yeah, there's a gene moving machine if I've ever seen one.
Oh yeah. So she actually made one initially, and you can see the smaller one. It just wasn't right. She didn't have the right number of subunits in the capsids and so forth. So she made number 2, and this is number 2 here.
Wow.
I would love to buy this, but you can't buy it.
I see.
But she does have an Etsy site. You can see here there's a link for her Etsy shop, and Gwen Beads on Etsy. Most of them have some kind of symmetry to them. They're a bit different than your normal beaded things. But, none of them are viruses. Here, this one is slugs in love, right?
There you go.
That's pretty cool.
Nice. Yeah. Maybe
looks kind of viral in a way.
True. Yeah.
So if you like beads,
surface.
that's pretty cool. I have a bead thing here. I'll show it to you.
Yeah. A lot of virus-related artwork there at the incubator.
So I have this model of poliovirus, which I have the genome is beaded here. And
That's
so
cool.
here's the genome. So this was made for me by Ann Palmenberg many years ago. Yep. These are beads, and there are 4 different beads here making up this genome, and there's 7,440 of them.
That's amazing.
4 different colors, and I'm going to show you, for example, here's the 3' end of the genome, right? What do you think red bead is, huh?
I'm guessing the admin.
Yeah, because that's the poly A tail right there at the end there. So she took the sequence and assigned a different color to each base, A, C, G, U, since this is RNA, and put them all together. And then at the other end of the genome, which is the 5' end, we have the viral protein VPG as a big bead.
Great. That's great. Yep. I love it.
So this is 7,440
Wow.
bases in length, and what I do is I stuff it in this capsid model here, and you know if you stuff it all in and try and get it out, it takes a long time to get it out
Hmm.
because
Hmm.
it's all tangled. But in a cell, when this virus infects a cell, it comes out, in nanoseconds. So something
Exactly.
magical is going on. Anyway, so this is a great teaching tool
Very
because
cool.
it's almost to scale actually
Yeah. Yeah.
for the virus particle.
Yeah. And Anne Palmenberg has sort of an indirect hand in the origin story of TWiEVO. So Anne is on the faculty of biochemistry
That's
at the
right. That's
University
right.
of Wisconsin-Madison. And so, Vincent, you mentioned at the top of the hour that we met at the American Society of Virology meeting when you were president of ASV. I was honored to receive the Anne Palmenberg Award for junior investigators at that meeting, and that Anne sponsored. And then she also gave you that bead. I mean, it's wild to think about, right? So in those like 7,000 beads, add another 20,000 beads, and you've got the genome of coronavirus, of SARS-CoV-2.
Yep.
And so, how consequential can a bead, a string of beads that you hold in your hand, be when it's sort of packaged up, and
Yeah.
that magic happens, where the viral machinery that it encodes comes, sort of breathes to life in an infected cell. All of the knock-on consequences that can have. It's really the power of viruses.
Um, so anyone wants to make a SARS-CoV-2 genome,
Yeah.
I'll pay you for it if you want to do
Yeah.
it because I'd like to have it here. I've got
Yeah.
some SARS-CoV-2 particles here, and would like to have that. I have a polio virus here. You can just see it
Mhm.
up there. That opens up. I
Yep.
could put another polio genome in there. I mean, it is rather
Cool.
tedious for you to do this, but
It's about
it's
28,000 beads. So that's going to,
a
take
lot
a
of
little
beads.
effort.
Um, a couple more comments here. Let's see. Um, there was okay, Mary Shelley. So could TWiEVO? Well, that says could TWiEVO cover the cell article, the genetic changes
Mm.
that shaped Neanderthals,
Mm.
Denisovans, and modern humans? I haven't seen it, but maybe we'll take a look in the
It's a good
future.
idea. It's been a minute. So I've dragged us, into the forests and ponds for the last few episodes. So we've been talking about things that will live in soil, fungi, worms, ants, and in today's episode, fish and eels. It would be, I think, we're due for a discussion on humans as
Sure.
well. Good
Okay.
idea. Yep. Thanks, Les.
Um, TB is from Poland. Best regards from Poland. Thanks for the
Great.
Kaprowski polio vaccine. That's right. Hillary Kaprowski
Hmm.
um, came from Poland. He ended up in Philadelphia. Made one of the two polio vaccines. Not been used, but he did make it and made a lot of contributions. So welcome.
Hmm.
And Sean says environmental DNA is totally new to me. Yeah, eDNA is a thing
Yeah,
for sure.
that's right.
And Farzad is from Iran. Welcome.
Right. Fantastic. Welcome.
Tremendous. I think we've had a few people previously from Iran and
Hmm.
maybe Iraq, but we'd love to have you. You came kind of late here. Sorry you missed it, but it's all recorded and you can always go back and listen to it. It'll be on YouTube.
Yep.
And one last time, let me remind everyone like we went to the trouble of making two nice pictures
That's
of
right.
us. So please come. We're going to have TWiEVO 100 live at the Science Mill in Johnson City, Texas, at about an hour outside of Austin. And that's going to be Sunday, March 10th at 5 p.m. If you want to come, we're going to have food afterwards, so you can hang around and meet us. You show up just before 5. We're going to start around 5. RSVP at microbe.tv. We also are going to do a TWiV on the Monday, which would be the 11th. And I think that's around 11 a.m. We'll have an invite for that, and I'll show that tomorrow on Office Hours live stream. So
Microbe TV tour of Texas.
we'd love to have you. Yeah, Microbe TV. What does it say on this?
Well,
It doesn't. There's
well,
supposed to be another.
you've got more stops on the way. This is just one of at least 3 that you'll be doing:
Yeah,
South by Southwest, TWiV, and then
and
TWiEVO
TWiEVO.
100.
Yeah, it's always great to combine stuff.
Yeah, yeah,
I love that.
really fun. Can't wait. So if you can't make it in person, join us on the live stream. Hopefully, we'll see how our AV technical skills are, but we'll hope to take comments in real time as usual. I'm excited already. I've got a pick of the week in mind that will be a video presentation that I'm hoping to pull off. We'll see.
Oh,
I'm
cool.
probably making promises I can't keep, but it'll be fun. We'll be at a place that celebrates science 7 days a week, and we'll be celebrating science in that space as well.
Space
100
that celebrates
episodes
science. Yes,
of
very good.
TWiEVO. Yeah.
So tomorrow we'll have office hours. I have yet to put the thumbnail up, but I will do so today. That'll be me and me alone, and we'll talk about viruses. I'll give you a little mini lecture at the beginning, and you can throw your questions at me and try and stump me, which is not very hard. So thanks for that. All right, that'll do it for TWiEVO 99, which, as Nell says, we cranked up the number of episodes just so we could have 100 when I'm in Austin, in a week and a half or so.
Yeah.
I want to thank everybody for coming today. Appreciate it. In these last few minutes, please hit the like button. There were 55 of you and only 32 likes. Hit the like button. It can help other people discover our program as well. I want to thank our moderators today. We had Les, we had Barb Mack, we had Andrew, we had Steph SF, and Vanity Nutrition. Appreciate you all coming.
Thanks, everyone.
You can find the show notes for TWiEVO at microbe.tv/TWiEVO. If you enjoy our work, we'd love your support. You can go over to microbe.tv/contribute. Our company, Microbe TV, is a nonprofit. So in the U.S. anyway, your contributions are federal U.S. tax deductible. Nels LD is at salvolution.org. And on Twitter, he's L Earlybird, E-L Earlybird. Thanks, Nels. Good to see you.
You too, Vincent. Can't wait. For less than 2 weeks, we'll be in vivo again for TWiEVO 100.
TWiEVO in vivo, folks. How about that? I'm Vincent Racaniello. You can find me at microbe.tv or here at the Incubator, which is in New York City. If you're visiting the city, we'd love to have you visit the Incubator. You can send an email to incubator@microbe.tv to set up a time when you want to come. By the way, the music you hear at the end, and I'm going to start that now.
Cue it up.
Is by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back, in a couple of weeks, right? Less than 2 weeks. Till then, be curious. Thanks, everybody. Bye-bye.